Hello everyone, this is our presentation on Single Molecule Imaging Uncovers Rules Governing Nonsense-Mediated mRNA Decay by Huoc et al. This was done by Ahmad Ali, Tim Tang, and Grace Zavada. Since we are doing a video, it will work a bit differently from the usual live presentations. I recommend opening up a word processor of your choice, noting down the slide number and time of the place where you have a question, and then asking it when we stop at regular points. Without further ado, let's begin. This technique is a modified version of a new technique called SUNTAG fluorescence labeling. The way it works is a reporter mRNA encodes an array of 24 SUNTAG peptides near the 5' end of the coding sequence so translation can be monitored, while the mRNA itself is monitored by 24 binding sites on the pp7 bacteriophage coat protein, called PCP, within 3' UTR. When translation gets going, SUNTAG peptides recruit GFP-tagged antibodies called scFv-GFP that fluorescently label the protein. Our reporter mRNA is fluorescently labeled and tethered to the plasma membrane via PCP-mCherry-CAAX. That's the fluorescent protein embedded in the membrane that binds to the 3' UTR hairpins that the researchers introduced. The foci initially appears red before translation begins. As the nascent proteins is introduced and the GFP antibodies are recruited, the foci turn yellow due to a mixture of green and red fluorescence. When mRNA is cleaved, the two foci appear together, the red and the green, and the researchers mentioned that tethering improves visualization without affecting translation or the NMD. You can see all of this happening in Figure 1a. Now we need to get some NMD reporters mRNA in. To do this, the researchers introduced the sequence encoding the model NMD target, triose phosphate isomerase, or TPI, into the reporter, either with the native stop codon, TPI-WT, a PTC at amino acid 160, TPI PTC 160, or at amino acid 1, TPI PTC 1. A sequence encoding BFP was included upstream of TPI to confirm expression of the reporter. The reporter was placed under the control of a doxycycline-inducible promoter. Figure B summarizes all this. Now that our NMD reporter is ready, we needed to do some reporting in real time. To do this, the researchers created some human U2OS cells. We're not sure why these cells specifically, but they were expressing TPI-WT or TPI-PTC160 reporters. This was done by transfecting the cells with the respective reporters' plasmids. The cells were observed using time-lapse spinning disk conofocal microscopy with 30-second time interval. Cells that expressed the TPI-WT reporter displayed red mRNA foci in the cytoplasm within 15 to 30 minutes of transcription induction by doxycycline addition. Most mRNA initially appeared without the associated green translation signal, but rapidly initiated translation which remained for the duration of the experiment. However, the green and red foci associated with individual translating TPI-PTC160 mRNAs often separated rapidly after translation had initiated. Figure C points this out. By the way, the researchers made a video of all this happening based on their real-time imaging technique, so here it is as well. So initially, I had planned on going through the entirety of this section, sentence by sentence. I even wrote up an entire script and then nixed it, because the remaining stuff is just the researchers now going over experiments from the supplemental material, and it's laid out in such an odd and confusing way that I'm just skipping to the end. So what is the researchers' conclusion from doing all this? What was the point? Apparently, it was to show off that the researchers could monitor NMD of individual mRNA molecules from start to finish in real time. No, seriously, this whole section, the one that kept leading me around in circles, was a big flex. I ain't even mad, that's pretty cool. Anyway, here's our quick summary on the section then. The researchers created a new way to look at NMD in action by modifying a SUNTAG fluorescence labeling approach. SUNTAG normally works by coding for SUNTAG peptides fused to the 5' end to monitor translation. Upon translation, GFP-tagged antibodies labeled a nascent protein. The mRNA is monitored by pp7 bacteriophage coat protein, which uses hairpin binding sites on the 3' UTR. PCP-mCherry-CAAX is embedded in the plasma membrane, labeling the mRNA and tethering it to the plasma membrane to improve visualization. And finally, the NMD reporter mRNA has additional modifications in the form of triose phosphate isomerase, modified with 2 additional PTCs in exon 1 and 5 respectively, called TPI-PTC1 and TPI-PTC160. BFP upstream of TPI is used to confirm expression of the reporter. On to an actual application of the new tech and not just showing it off. The researchers wanted to know what was going on with ribosomes and NMD. They mentioned prior models describing what was thought to be going on. One model described NMD as occurring on CBC-bound mRNAs during translation within the first few ribosomes. Another model describes potential for NMD to happen at any point of translation, but later mentions that many of these models are rather inaccurate, so they proposed their own model and tested it. The proposal was that, quote, the cleavage rate reflects both the time of arrival of the first ribosome and the probability that a ribosome will induce NMD upon translation termination, end quote. Basically, that any ribosome can induce NMD and not just the ones that are present. To study this, the researchers needed to know 2 things. The first was the probability of a ribosome terminating translation through NMD, and the second was how often ribosome termination events occur. From their supplementary data and prior research, the researchers calculated that the ribosome termination events occur at a rate of 3.2 ribosomes per minute. This was based on, quote, on the translation elongation rate and ribosome occupancy of TPI-PTC1, end quote. Basically, they did another experiment where they looked at NMD and TPI-PTC1 correlation. Based on this and the data shown in Figure 2a, their experiment seemed to support their model over the first ribosome model. As you can see, the blue line represents how the first ribosome model would have functioned, the dotted line represents how the actual data happened, and the red line represents the any-ribosome model. This revealed that 90% of the mRNA were degraded in a population that was rapidly decaying. They also found that each ribosome had an 11% chance of setting off NMD. Based on this, the researchers hypothesized that any terminating ribosome has an equal chance of doing NMD, and their model was on the right path. The researchers wanted to see which ribosomes were inducing NMD, so they created a new TPI-PTC1 reporter called HP21-TPI-PTC1. This one had a strongly reduced translation initiation rate due to an added 21 nucleotide hairpin in the 5' UTR. This helped them to count individual ribosomes translating the reporter mRNA. In Figure 2c, using GFP to measure intensity, the researchers noted clear peaks of GFP signals that lasted minutes. They were interspersed by periods lacking the detectable GFP signal. Since each peak corresponds to a ribosome, they were able to precisely determine the number of ribosomes that initiated translation. Skipping past their references to supplemental data, the researchers found that a median of 8 ribosomes translated the HP21-TPI-PTC1 reporter before NMD was triggered. Figure D shows the results. Based on their methods, the researchers concluded that this corresponds to a probability of 0.01 plus or minus 0.03 of inducing NMD for each terminating ribosome, which was close to the earlier 0.11 or 11% probability from earlier. Alright, now to hand it off to Tim, who's going to talk about how NMD does not occur preferentially on CBC-bound mRNAs. Good luck, Tim.
Alright, in part 3 they wanted to look at the effects of CBC and EAF4E on NMD efficiency. But before that, here's a quick review on the cap binding complex and EAF4E. CBC is the nuclear cap binding complex which binds to the 5' cap of the mRNA. It protects the cap as well as assisting in nuclear export of the mRNA. The CBC is supposedly replaced by the cytoplasmic cap binding protein EIF4E, which is called the eukaryotic translation initiation factor 4E. EIF4E also binds to the 5' cap and assists in the binding of the mRNA to the ribosome in the early stages of translation initiation. The hypothesis for this section is that if NMD occurs preferentially on CBC-bound mRNAs, then the rate of NMD decay should be reduced over time as more of the CBC is replaced by EIF4E. Looking back at figure 2a, the authors noted that the cleavage rate for all three of the reporters, TPI-PTC1, TPI-PTC160, and beta-globin PTC39 was fairly constant, which kind of contradicts their hypothesis. They believe that CBC-EF4E exchange occurs approximately after 10 minutes, but here we see that the mRNAs rapidly decayed in under 10 minutes. So, to determine whether or not the decay rate remained constant over a longer period of time, they partially depleted UPF1 in the U2OS cells expressing either TP1-PTC160 or beta-globin PTC39. UPF1, also known as frameshift protein 1, is the RNA helicase that assists in NMD by removing mRNA-associated proteins. Partial depletion of UPF1 was done using siUPF1. This was done since the authors believed that partial depletion should reduce the rate at which NMD occurs, and thereby giving more time to observe translation. What they found here was that the rate of decay still remained constant over the entire 40 minutes of the experiment. Next, they looked at whether NMD efficiency was affected by the translation initiation rate. They did this by replotting the cleavage kinetics from HP21 TPI PTC1. And again, the rate of cleavage was constant throughout the entire experiment, which suggests that the translation initiation rate does not affect NMD efficiency. Another thing they looked at was the efficiency of NMD while mRNA is bound to either CBC or EF4E. To do this, the authors needed to determine the exact moment the switch from CBC to EF4E occurred. They used an inhibitor of EF4E called HA4EBP1 in order to inhibit EF4E dependent translation. HA4EBP1 is a variant of 4EBP1, which is a protein responsible for repressing translation. Normally, 4EBP1 binds to EF4E and represses its activity, but phosphorylation of 4EBP1 releases EF4E, and that re-enables its ability to activate translation. Since HA4EBP1 is a non-degradable, non-phosphorylatable variant, it can be used to repress translation in cells. The authors overexpressed HA4EBP1 in cells from before that were expressing SCFP, SFGFP, PCPM cherry CAX, and then observed the fluorescence. They saw that the control cells stably displayed a greenish yellow fluorescent signal throughout the entire experiment. Meanwhile, the cells overexpressing HA4EBP1 displayed kinetics similar to the control before the translation initiation was significantly reduced or terminated around the 5-minute mark, which is shown by the appearance of red fluorescence. They also looked at determining the precise moment of translation initiation, which was done using their own program called RiboFitter. And RiboFitter is a program that's used to determine the precise moment at which each ribosome initiates translation using fluorescence intensity. And so each peak in fluorescence represents translation initiation by a singular ribosome. Here they found that the control cells had an initiation rate of approximately 2 to 3 ribosomes per minute, which was constant throughout the experiment. The cells overexpressing HA4E-BP1, on the other hand, had similar rates before suddenly stopping for a period, followed by a resumption of initiation. The author speculated that the burst in initiation after the halt was very likely due to dissociation of HA4E BP1 from EF4E, or the binding of a new EF4E that was not associated with HA4E BP1 to the cap. The initial initiation was likely due to the HA4E-BP1 cells translating CBC-bound mRNA, and that translation initiation halted once the transition from CBC to an inhibited EF4E began. So to conclude this section, they found that the rate of decay remained constant throughout the entire experiment, and that the rate of translation initiation did not affect MND efficiency. Most importantly, since the switch from CBC to EF4E occurred within the 40 minutes, they concluded that NMD does not prefer either CBC or EF4E bound mRNA. One of the previous experiments demonstrated that their real-time imaging technique could be used to analyze the cleavage rates between different mRNA reporters, but now they want to look into the mechanisms behind it, such as cleavage rate and NMD resistance. The first thing they looked at was how nucleotide sequences downstream of the PTC can affect MND efficiency. This was done using another standardized reporter containing the gene KF18B, a PTC, and various intron cassettes, which were located downstream of the PTC. They found the different cassettes produced significantly different cleavage rates as shown in figure C, as well as affecting the fraction of mRNA degraded. So next they selected 3 reporters with widely varying cleavage rates to confirm that the cleavage is indeed caused by NMD. This was done like before using SI-UPF1 to confirm that these reporters were being degraded by NMD. Here in figure 4D, it shows that UPF1 depletion results in a greater fraction of intact mRNA over a period of 40 minutes compared to the non-depleted cells. And next they tried swapping introns within the cassettes to see if the introns affected NMD efficiency. Figure 4E shows them switching the introns of the CD63 and MITD1 cassettes. In figure 4F, there appears to be no major differences between swapping the introns, and since they saw no differences, the authors believe that the introns did not affect NMD efficiency, and this might suggest that the exon sequence directly downstream of the PTC may influence NMD efficiency. Finally, they wanted to make sure that the mRNA reporters that remained encleaved were not later degraded through exonucleolytic decay. Here they found that none of the mRNA reporters experienced further decay 40 minutes after translation initiation. This likely meant that they did not experience any decay by an exonuclease. And to finally finish this section off, they concluded that the introns did not affect NMD, but rather likely the exons downstream of the PTC did. Additionally, they also concluded that mRNA not degraded by NMD were also not degraded by nucleases.
Wanting to build on previous studies with their new imaging technique, the authors decided to look at the effect downstream intron distance had on NMD efficiency. 2 previous studies showed long distances between a PTC and intron decreased efficiency. However, this was also the case if the PTC was too close to the intron. From this, they hypothesized the latter was a result of the ribosome kicking the EJC off the mRNA. Remember, the EJC is bound at an exon junction and communicates with ribosomes using NMD factors to target it for destruction, so it is critical to the NMD pathway. To further prove and validate the effect distance has on NMD, the same KIF18B PTC TPI reporter used earlier had linker sequences of 100, 175, 250, or 1000 nucleotides incorporated between the PTC and exon 6. The composition of their control, the gene with no inserts, is shown at the top of figure 5A. Below is the modified reporter with the linker insert. A second assay was set up to investigate how small distances between a PTC and intron affects NMD. This was done by inserting PTCs into the TPI gene at 73, 52, and 40 nucleotides upstream of intron 6. Looking at larger distances, they found there was an increase in uncleaved mRNAs as the linker distance increased after 263 nucleotides. This was represented in figure 5b, where the lower the curve, the more NMD activity occurring. This was all measured by watching the movement of foci mentioned at the very beginning. However, figure 5c shows that as distance from the intron increased, a decrease in the rate of NMD occurred. So although a 188 and 263 nucleotide distance resulted in a higher amount of cleavage, having no linker made getting the job done much faster. Doing this also presented the results in figure 5d that the fraction of NMD-resistant mRNAs was not substantially insignificant. Translated, that means there was a slight change in NMD-resistant mRNAs, as shown in D, but not significantly enough for them to touch on. These tests were also performed on another different reporter where similar results were produced. Moving on to the second set of results, they found decreasing the distance from a PTC to an intron produced a similar effect. This distance shortening led to a strong decrease in NMD activity, which was attributed to an increase in NMD-resistant mRNAs, shown in figure 5d. This data backed up their idea that the ribosome moving bumped the EJC off and stopped NMD from occurring. They also found that while NMD activity is reduced, it is not zero, and NMD was still able to degrade a low amount of mRNA, meaning not all mRNA can have their EJCs bumped off.
Now a quick summary on the section before taking questions. The researchers found there are distance requirements to obtain maximum efficiency of NMD, and also NMD-resistant mRNAs occur in every population.
Next, the researchers moved on to looking at the effect the number of introns both upstream and downstream of the PTC have on efficiency and NMD-resistant mRNAs. They used intron cassettes already present in the gene to lengthen their experimental copy, so as to avoid introducing any more variation into the gene and keep things consistent. Looking at introns introduced downstream first, they used the KIF18B PTC-CD63 gene as their reporter. A second copy of the CD63 intron was inserted downstream of the PTC, shown in figure 6a. This introduction led to an increase in NMD activity and decrease in the fraction of uncleaved mRNA, represented in figure 6b. Taking things a step further, the authors used previously sequenced cancer cells to look at how nonsense mutations affected mRNA levels. Using this cohort, they found NMD rates lower in PTC-containing mRNAs with only a single EJC downstream. This data was collected into figure 6c. To explain these results, they came up with two different models. Their first model was the hypothesis that the increase in intron cassettes would increase the number of EJCs present on the mRNA, as one would be added for each new cassette, increasing the chance of a terminating ribosome interacting with an EJC and inducing NMD. An increase in efficiency from this could only be affected by downstream introns. The second idea was NMD could be increased through altered mRNA processing. They gave the example that altered processing could enhance an NMD stimulating protein being loaded onto the mRNA. This model means introns placed upstream of a PTC could also affect NMD efficiency. They created a second experiment to test these models using the KIF18B PTC-NUBP2 gene, reasoning that it had displayed a moderate cleavage rate earlier, though not indicating when, to allow for easier detection of increases and decreases in NMD. Figure 6e showed both upstream and downstream intron insertions increased efficiency, done against a reference of no introns included. Similar results were found in the cancer cells mentioned earlier. Efficiency was boosted the most with upstream introns when 4 or more cassettes were implanted, but did not contribute when 2 or more were inserted downstream. They believed this showed both their models were correct and NMD is increased by a higher concentration of EJCs being present on the mRNA, as well as the change in the processing.
To conclude this section, the authors found upstream and downstream introns had positive effects on NMD efficiency, so more is better as long as it doesn't mutate. Any questions?
For the final portion of this paper, the authors decided to take a close look at the rate at which XRN1 exonuclease degrades the 3' membrane-bound fragment of mRNA after it has been cleaved. Studies have looked at this before, but they wanted to use their new technique to look at it in a live environment. To perform this experiment, the TPI PTC 160 mRNAs were imaged under high temporal resolution, monitoring the time it took for the red fluorescence to disappear. To test the processivity, they reduced the levels of XRN1, hypothesizing that if XRN1 is highly processive, the start of decay would be slowed but the rate would remain the same, while a decreased rate and faster start would indicate weak or non-processivity. Figure D is a mock-up of what the processivity would look like under normal circumstances. The black line represents what normal processivity under normal conditions should look like. The blue and red lines are under reduced XRN1 levels, where the blue line has a shallower slope because of its increased rate, and the red one does not begin decay until much later. Their results pointed to the model of high processivity as can be seen in Figure 7c. The fit matches the red line from the previous slide. Figure E shows the experimental results against a standard called SI-MOCK, which demonstrate a clear increase in the time from cleavage to start of decay, but Figure F shows its decay time was nearly identical to the standard. They then moved on to test processivity more precisely by creating a new reporter using TPI, shown above, and adding 48 times more of the pp7 cherry array that anchors TPI to the membrane and gives it its fluorescence. By adding these two extra anchors and fluorescence at separate points, the rate can be closely monitored by the disappearance of each tag. The fluorescent remains at 50% during the decay of the 2.6 kilobyte linker. This more precise method partially confirmed their theory of high processivity. However, it also showed new results, displayed in Figure I. About 60% of mRNA underwent rapid degradation of the linker and agreed with their previous results, but the other 40% showed a much slower degradation of the linker. This is split up between figures J and K as the number that have not yet started decay over time and the number of mRNAs left with max fluorescence, respectively. In an attempt to explain this, the authors came up with the hypothesis that the rapid degradation of the mRNA linker was caused by a single XRN1 binding event, whereas the slow degradation was caused by a necessity for more than one mRNA binding event to happen. They cite the fact that the reduced concentration of XRN1 did not substantially affect the fraction of mRNA molecules that were rapidly decayed, but did increase the decay time of slowly degrading mRNAs. This suggests the increase in decay time is not from reduced efficiency of XRN1, but is because the XRN1 molecule fell off of the mRNA and then had to wait for another one to rebind. They assume this is because XRN1 degrades much faster than the ribosome can leave and is knocked off as a result.
There isn't much here to summarize, but the main points gleaned from this experiment are the high processivity of XRN1 in vivo, and because of this, it can sometimes fall off the mRNA before finishing degradation. Any questions? Before concluding this presentation, we have some criticisms we would like to address. First, we believe the authors could have had more standard experiments present on their figures. An example of this is in Figure 5F. They could have included the TPI-PTC wild type or PTC1 signal they had obtained earlier. Instead of using a program to calculate the results for Figure 3H, they could have performed the experiment with the reporters. Some of their figures are very hard to understand, and overall, the paper is laid out in a disorienting way, full of tangents to the researchers' other experiments and supplemental data before coming back around to discuss their main points. They also don't appear to have an objective and seem more interested in trying out their new technique. They don't really give any suggestions for applications of all this data, but
other researchers have been looking at the link between NMD and diseases such as ALS. One particular paper, linked in the slide, found that by overexpressing NMD factors, they were actually able to decrease the level of toxicity caused by a repeat expansion in the C9HRE gene. This gene is the most common cause of ALS and frontotemporal dementia. In the opposite direction, researchers studying cystic fibrosis look at inhibiting NMD as a way of stopping the truncation of a protein that interferes with the cystic fibrosis transmembrane conductance regulator. This interference leads to a dysregulation of fluid in the lungs and results in cystic fibrosis. Inhibiting NMD for this molecule would lengthen it and hopefully stop it from interfering with the CFTCR. Back in the section discussing the effective distance between intron to PTC, it concluded by stating there was a population of NMD-resistant mRNAs that could not have their EJCs bumped off, that was present in all three of their experimental genes. However, the experiment was performed on cloned cells that should be hypothetically identical. The authors or other researchers could try and find out what mutations were causing this. What is happening where the EJC is binding that allows it to stay when most of the time it is displaced? This would be trickier to capture as this information is known because the mRNA strands are being degraded, but also using that to their advantage, they could know the exact moment of cleavage and attempt to fix cells there to try and preserve the cleft mRNA for sequencing. They also found that without XRN1, NMD cannot occur and decrease the amount available to hold off degradation.
So in conclusion, this paper demonstrated a new assay to visualize NMD in real time. This assay revealed many aspects governing NMD. It showed that NMD does not discriminate between CBC or eIF4E bound mRNAs, and that it occurs with equal probability on each round of translation. This deficiency depended on the sequence downstream of the PTC, the distance of the PTC to intron, as well as the number of introns around the PTC. And finally, they showed that the exonuclease XRN1 is highly processive in vivo in the decay of the 3' mRNA after cleavage.
Oh, it's a setup, it's a setup, it's a setup, there's an oh, it's a setup, it's a setup, it's a setup, there's an oh, it's a setup, it's a setup, it's a setup, there's an oh, it's a setup, it's a setup, it's a setup, there's an oh, it's a setup, it's a setup, it's a setup, there's an oh, it's a setup, it's a setup, it's a setup, there's an oh, it's a setup, it's a setup.